Welcome to the course of Plant Developmental Biology. So, in previous class we were discussing the reverse genetics based approaches, and we have already finished how to analyze or how to monitor gene expression pattern. And then now in this class we are going to discuss about how to functionally characterize a gene. So, to functionally characterize a gene, which means that you want to know what is a specific function of a gene in a particular developmental process. So, how will you know that? So, one approach is always to know someone's function is loss of function. So, if you somehow disrupt the gene and then see what kind of developmental defect you see when you make a loss of function of a gene, and this will tell that for what function your gene is necessary. And then another complementary approach to validate it or to understand the function of a gene is the gain of function. So, normally let us assume a developmental gene is expressed in a particular tissue, but it is not expressed in some other tissue. Then if you take this gene and ectopically express in the tissue where it was not present normally, and then you can look that tissue, can it reinitiate some developmental program? And if this is true, then we can say that a particular gene is sufficient for a particular function. So, these are the two major approach which we take to understand function of a particular gene in development. So, we will take one by one. So, first we will take loss of function, and here there are two way to do it. Either you knock out the gene or you knock down the gene. In knockout, what you basically do, you totally make a null mutant where the gene is totally disrupted. In knockdown, you are reducing the expression of a particular gene, but it is not null mutant. And in gain of function, there are two way to do that. One which I told is ectopic overexpression or ectopic expression. So, you express this gene, you change the promoter, you use some broader promoter to express this gene in a tissue where normally it is not expressed. And second method is activation tagging, which we will see in later on. So, first we will take the loss of function, how to generate knockouts. So, if you look the first two approaches, this we have already and also discussed in the forward genetics based approaches, which is to generate insertional inactivation lines for a particular gene. So, one was the tDNA insertion, second was the transposon mediated insertion. But the difference between forward genetics and reverse genetics is that in forward genetics we were not fixing the gene, we were fixing the function, or we were fixing the phenotype, or we were fixing the biological process, and then we are trying to look for tDNA insertion lines or transposon insertion lines, and then try to map that which gene it is. But for in the reverse genetics, what we do that now we know a specific gene, now we know that this is the gene and I want to understand what is its biological function. Then you can specifically go and look for that gene, is there some tDNA insertion lines available for that gene or transposon lines available for that gene or not. Another and now very widely used method is genome editing. So, here you are basically editing at the genome level the gene, and then you are generating knockouts or loss of function mutants. So, first we will take the insertional inactivation lines. So, as I said that the initially people have done at a very global level, they have generated a large number of tDNA insertion lines or transposon insertion lines, and those lines has been deposited to some databases. So, for example, there are already so many databases or so many laboratory or so many projects, they have generated a large number of tDNA insertion lines or transposon insertion lines, and these all mutants has been preserved and protected. And what you can do that you can choose any of the mutant, you can request, you can order, and you will directly get those mutants for your detailed functional analysis. So, there are many large number of this kind of collection of mutants are available. I will take one of the examples. So for example, this is tDNA Express, or this is a Salk Institute. They have generated a large number of tDNA insertion line. What simply you have to do, this website is available. You just choose your gene, put the gene ID, search with the gene, search with the chromosome, search with the some of the name or anything, and then they will show a list of insertion lines available for that particular gene, and you can choose. Sometime what happens that you might have more than one insertion lines in your gene, and then you can look, you can define where you want to have the insertion. For example, if you look this gene, you can have so many insertion. You can choose that I want one insertion line in exon 1, I want one insertion line in exon 2, and then you just take these numbers and request from the database, and these seeds, the mutant Arabidopsis seeds, will be available to you, and then you can get these mutant or knockout lines for your detailed functional characterization. And the first thing what you do, if you get this kind of insertion line, you first establish that actually there is insertion, first thing. Second thing is that what is the genetic pattern? Is it insertion is homozygous, both the alleles are disrupted, or it is heterozygous, only one alleles are disrupted? How you can do? This is very easy, since you know the site of insertion, predicted site of insertion, you know the sequence of tDNA, you know your gene ID, so you know essentially the sequence of your gene. You can design a forward primer here, one reverse primer here, and one primer you can design in the tDNA, and then you can do some PCR. If you do with this genomic DNA forward and reverse primer, if there is insertion, the insertion line will not give it, because the fragment size is very big. You will not get the PCR amplification. So if you have a homozygous line, in homozygous line is basically your both the genomic locus has the insertion. Then if you use LP and RP together, you will not get a band here. But if it is wild type or heterozygous, in heterozygous one locus has the insertion, another locus does not have insertion. Then from the locus where you do not have insertion, you will get this band. So basically your wild type and heterozygous both will keep band with the LP and RP. Then you choose your LP with this or RP with this, depending on in what orientation your tDNA is. And then if you have a heterozygous, you will have a kind of band here, and then if it is wild type, you will not have this tDNA specific band. So this is very important for you to establish that what is the insertion status of this tDNA in your gene. And another important thing what we try to do is that we always backcross, because since all this has been generated through a random insertion, there is a possibility that in your plant there are more tDNA insertion site. So you might have an insertion in your gene, but there might be some other insertion somewhere else in other genes, and that you can remove by taking these lines, crossing with the wild type, and then going to the F2 generation and again screen for homozygous. And then once you have established this line, you can take these lines for detailed functional characterization or study the developmental defect in this tDNA or loss of function lines. Another method, which is genome editing. So in genome editing there are many techniques available which you can use to edit the genome, ZFN, then TALEN, but recently CRISPR-Cas9 has been used very widely and very extensively these days. So we will see how this is being used to edit the gene. So essentially what is this CRISPR-Cas9 based genome editing tool? So here what we are using a bacterial endonucleases, which is RNA guided endonucleases, that is Cas9. So and then what you do, simply you produce a guide RNA and you produce Cas9 protein in the plant. This guide RNA you can target to your sequence wherever you want. So you can design a very targeted guide RNA, and then when you express this guide RNA along with the Cas9 protein, so maybe that will be clear here. So what you do, let's assume this is your gene and you want to edit this gene, you want to disrupt this gene. What you can do, you can choose a target sequence typically here, and there is one requirement which is called PAM sequence. So you can target your region which is near to the PAM sequence, and you can generate a guide RNA which is specific for this sequence, and then you clone this guide RNA in a vector. In the same vector you can clone Cas9 endonuclease protein, and then when you take this and make a transgenic plant, you both the partners will express there. Then what will happen, this is a guide RNA, this is Cas9 protein. When Cas9 protein will bind with the guide RNA will target this endonucleases in the sequence specific manner, and when this endonucleases is recruited on the genomic copy, it will make a double stranded break in the genomic copy, and then this break has been considered as a damaged DNA. An endogenous mechanism which is called non-homologous end joining mechanism will be activated in the plant, and it will fill this gap, it will repair here. But during the process of this, since it is end joining mechanism, there is a possibility that you can generate a deletion, you can generate an insertion, you can disrupt the coding region, and that is how you will have a genomic disruption of a particular gene. So this is some of the method which is being used for making the knockout lines. Another mechanism what we are using to study the loss of function is knockdown or down regulation. So here we are not disrupting the genomic copy, but we are regulating after the transcription. So we are reducing the expression level either at the protein level or at the transcription level, and there are some of the techniques which is being used for this function. One is the antisense RNA technology. This was one of the pioneer technique which was being used, but these days there are many more techniques. So then second method is double stranded RNA interference or artificial microRNA. So in antisense RNA, what happens that if you have a gene and you know the gene sequence, you just take the same gene or the gene specific fragment of the gene and clone in the antisense orientation. And then what are you expecting? So if it is a genomic copy of your gene, it will transcribe in this direction. But now you have generated another transcript which is complementary to this endogenous transcript, because it is antisense orientation, and then this transcript will go and it will bind with the endogenous transcript, and then this transcript either it will be degraded or it will be inhibited for protein synthesis, and you will have the phenotype. This is one example that there are few genes has been targeted through antisense RNA. You can see this is the promoter, and the genes has been, and the gene specific region has been cloned in the antisense orientation. And when you drive these genes, you can basically suppress the endogenous gene using this method. As you can see the phenotype, so this is control. When the genes has been suppressed, there are a lot of developmental defects, and when you check the RNA label or the transcript label, you can clearly see that as compared to controls in these antisense lines, there is a significant reduction of the transcript label. But there are something important. If you look this line here, almost down regulated, but these lines are partial down regulation. So this is not a kind of null mutant, but this is a kind of down regulation. So you can have here a gradient, or you can have some lines where you have near to the null phenotype, but some lines you might have which are weak lines, there the down regulation is not so efficient. Second method which is being used for down regulation is double stranded RNA interference technologies. This technology were developed from the endogenous mechanism. So in plants, siRNA and microRNAs are known as a well regulatory mechanism. There is a large number of small RNAs, and they are regulating expression of many important endogenous gene. So if you look at general mechanism, there are two ways: either you have a double stranded RNA or hairpin RNA. This will undergo the process of processing through dicer or ego mediated processing, then uploading. This I will not go in the detail, I am sure you would have already studied, and then it generate a very small 21 to 24 nucleotide single stranded antisense RNAs, and these RNA they can go and pair with the endogenous messenger RNA, and then they will either target that messenger RNA for the degradation, or they can inhibit protein synthesis, or they can do epigenetic regulation. Any kind of regulation is possible. So, to do that, if you want to use this technology, so what you can do, you can generate your own RNA interference construct for a specified genes. What you have to do, you have to simply mimic this hairpin generation. How you can do? You can do take a gene specific fragment and clone in sense orientation, and then put some non-specific sequence here or stuffer fragments here, linker fragment, and then you take the same fragment and clone downstream, but in opposite orientation. If this is in the sense orientation, this you put in the anti-sense orientation, and put a promoter here, drive this hairpin loop, and then what will happen, this messenger RNA which will make this kind of a structure. So, this region and this region will pair here, and it will generate a hairpin structure, and this will be endogenously processed, and then it will generate siRNA, and this siRNA specifically will target the endogenous RNA or messenger RNA against which it has been designed, and then it will down regulate the expression of those genes. There are plenty of examples where RNA interference has been used to down regulate the gene. Some of them I will show here. So, if you look here, two of the genes has been down regulated by RNA interference based approach. You can look here, this is northern blot. In control there is high level of expression, but in RNA interference line the level has significantly decreased, and you can see the phenotype, panicle phenotype. Similarly, this is a very important transcription factor MATS1, and when the RNA interference line was generated for MATS1, you can see this is northern blot. Again, in wild type you have a very high level of expression, but in RNA interference line the endogenous expression is almost null, whereas you can detect the hairpin structure which is basically bigger of the size. Not only that, if you go and do a very special northern blot, you can detect small siRNAs as well. So, these are the small siRNAs which is not present in the wild type, but it is present in the RNA interference line. So, this has been very extensively used to down regulate many of the genes. Then third method which is being used to make knockdown mutants are artificial microRNA. So, as I said, microRNA is a natural small RNA present in the genome of the plants. They are being processed through dicer and ago mediated mechanism, and then this microRNA they are going and targeting specifically some of the genes and regulating its expression at different level, transcriptionally, post transcriptionally, translationally, epigenetically, and then this technique was taken and being used to generate some artificial microRNA. So, basically what you do, that if you take a natural hairpin structure or the loop of a naturally existing microRNA, and if you can replace these core sequences, the microRNA core sequences which is basically showing the complementarity with the gene, and if you replace with your own gene of interest, can you down regulate it? And then this can be easily done by a combination of multiple PCR. So, for example, if this is your construct here, you can use some different set of primers. You can amplify this region from here to here, then you can amplify this region from here to here, and you can amplify this region from here to here. So, basically this red is your artificial microRNA sequence. Then if you use these three as a template together and use these two primers, so you can combine them in this way, and then this artificial microRNA you can clone under promoter and then generate some knockout line. And if you see here, this has been quite successfully used in some of the study. For example, if you take this example of Arabidopsis, in Arabidopsis this is leafy mutant. So, this is basically insertional mutant. So, if you look leafy 12, this has a significant phenotype as compared to the wild type, but if you target this leafy through artificial microRNA against the leafy, you can mimic the phenotype. So, your artificial microRNA here is as strong as the other mutants. Similar is the case if you look FT mutant. So, FT mutant, if you look the wild type, the plant is already flowered, it has achieved the reproductive stage, whereas this FT mutant has very delayed flowering. But if you generate again artificial microRNA against the FT, you can see that it is showing as severe phenotype as the other mutant. So, this tells that artificial microRNA is one of the very efficient way to downregulate genes in Arabidopsis. And similar kind of things has been used in other plant species. For example, if you take the case of rice, so here artificial microRNA has been generated for a couple of genes. So, this is SPL11, this is PD's gene, and you can clearly see this is the control and this is the RNA artificial microRNA line, and you can see a clear phenotype in artificial microRNA lines. So, basically, so through this study, either you go through the knockout based study or you go through the knockdown based study, what you conclude that you can generate a loss of function mutants, and you conclude that these genes are necessary for this particular function. Another complementary way, as I said, that is gain of function. So, first way that we can generate ectopic overexpression or ectopic expression construct. Here what you do, you use some broader promoter or a promoter which is expressed at the high level, and you can use either tissue specific promoter for the tissue where your gene endogenously does not express, or you can use a kind of general 35S promoter or ubiquitin promoter to drive your gene in the sense orientation. And then you can have high level of expression in the tissue where you do not expect your gene endogenous to present. And if you look the cases in this mutant, you can clearly say that this is your basal level of expression, but when you have overexpressed under 35S promoter, you can see that in most of the lines the expression level is very high as compared to the control, and you can see here the relative expression, and then this is correlated with the phenotype. So, this is and then if you go to the next generation, you can clearly see this is one of the line has been tested in the next generation. So, maybe here the lines is segregating. If you look this plant, it is not showing phenotype, but these two plants are showing the phenotype, and if you go and check the overexpression level, it clearly correlates. So, this plant which is not having phenotype does not show the overexpression as well, but these two plants which show the overexpression, they show the phenotype. Similar kind of analysis has been done for the transcription factor MAX1, and you can clearly see when MAX1 was ectopically overexpressed in transgenic rice, you can see a very significant phenotype in the panicle architecture and the spikelet development. And if you see the expression level, you can clearly see that in the control you do not have expression in this tissue, but these lines they have a very high level of expression. So, another way to generate a gain of function mutant is activation tagging. In activation tagging, basically what you do, you take a tDNA construct and you just put some multiple copy of enhancer elements. Here you can take 4x copy of the 35S enhancer element and then randomly throw in the genome, and it will go and integrate. But what happens that if it comes in a proximity with some of the basal promoter or some of the promoter which normally is expressed at the very low level, or normally it is not expressed, but now the promoter has got some of the enhancer element in the close proximity, and then the promoter will start activating, it will start very strong expression, and in that case what will happen that you will see a phenotype which could be because of the gain of function phenotype. These are some of the example. So, if you look here, there is a insertion of enhancer elements here, and if you look the result of this is that if you look this gene is more expressed in the lines insertion line than the control. So, if you look here and then you can see this, there are some of the phenotype of this line. So basically this is called enhancer tagging. You can look for many more genes and identify more activation tagging lines, and you can take them for the studying their function. So this is a typical way to functionally study gene in plant, but there is some problem. So let's assume that if you want to study function of some of the essential genes, what happens if you make a knockout or if you make a knockdown? If gene is essential, you face a problem of lethality. If it is lethal, if loss of function is lethal or gain of function is lethal, you will not have a transgenic plant, or you will not have a plant to study. In that case, how to solve the issue? Some of the genes are very important at very early stage during embryo development, so they could be embryonic lethal. Some of the gene might be not embryonic lethal, but they could be lethal at the seedling stage or at any stage. They complete their vegetative growth, but they cannot enter in the reproductive phase. So basically they will not make a fertile flowers, they will not make seeds, which means that you cannot go to the next generation. So if you want to study such kind of genes through the reverse genetics based approach, how to go it? In that case, what you have to do, you have to use a regulated gene expression based approaches. So you cannot take a gene, or you can down regulate all the time, or you can down regulate constitutively, or you can overexpress constitutively. If you do that, you will not be able to study them. Then what you can do, you can generate a construct, or you can make a strategy where you can specifically regulate a gene whenever you want, or you can have a kind of more temporal and spatial based regulation of your gene silencing or overexpression. So there are three ways how you can regulate gene typically. One is the spatial regulated construct. So what you can do, that instead of overexpressing a gene in all the tissues, you target overexpression in some of the tissue where you feel that might not be so harmful that it will have lethality. For such kind of things, you can use some tissue specific promoter, some known promoter which is expressed in a particular organ. For example, you can check in the leaf, and you can use some leaf specific promoter and express a flower gene in the leaf, and look, can it start some of the flower specific developmental program in the leaf. Second way of regulation, what you can do is the temporal regulation construct. Here what you can do, you want to express or down regulate a gene at a particular stage, not before that. For example, let's assume that down regulation of a gene X is embryonic lethal, which means that if you down regulate from day one, even embryogenesis will not complete, then you will not have plant. In that case, what you can do, and if you want to study the role of this gene maybe in the vegetative development or maybe in the reproductive development, then you do not need to down regulate or silence or overexpress this gene during the process of embryogenesis. So what you can do in that case, you can use some stage specific promoter to drive the construct, overexpression construct, RNAi construct or any construct, and then what you can do, you can skip your early stage of embryogenesis. So your embryogenesis will occur normally, then later on you just down regulate and see what is the consequence. Second is that you can even combine both, and you can generate a construct where you can do both together. So you can regulate a gene or a construct, overexpression RNAi construct, temporally as well as spatially. How you can do, we will take just one example of both the cases. For example, if you are studying a transcription factor, there is a system which is being very widely used, is a glucocorticoid receptor based system. So we know that transcription factors, they has to enter inside the nucleus for activating the gene expression, and if you somehow retain them in the nucleus, and if you can regulate their translocation between cytoplasm and nucleus, you can regulate their activity. And that is being done by fusing your transcription factor with a rat or mouse glucocorticoid receptor. So glucocorticoid basically is a steroid hormone in animals, and what happens that the glucocorticoid receptors, they are normally present in the cytoplasm when they receive signal through the hormones, and then what happens that they only then enter inside the nucleus, and go and activate their genes. So, if I take a gene, here is the example of MATS1. If I take MATS1 gene and fuse with delta GR and overexpress under any broad promoter, does not matter here, 35 as promoter, then what will happen? Under normal condition, I have a high level of MATS1 expressed, protein is already made, fusion protein, MATS1 delta GR fusion protein is already made, it is ready, but it is only present in the cytoplasm. Since MATS1 is a transcription factor, until and unless it will enter inside the nucleus, it cannot perform the function. Then if I take this plant and induce with hormone, which we are using here, dexamethasone, then what will happen? It will enter it in the nucleus and it can activate the gene. And this is very important technique which you can use to even distinguish between direct genes which are regulated directly by the MATS1 or the gene which are consequence of the MATS1 induction. So, if you combine this dexamethasone treatment with a protein synthesis inhibitor cyclohexamide, so let us assume if this is example of OSMGH3, OSMGH3 as I said earlier that this is one of the gene which is regulated by MATS1. And what happens when you treat with the dexamethasone, you can see at a particular time point the expression level of OSMGH3 is going up, which means that MATS1 is sufficient to activate expression of OSMGH3. But when you treat with the cyclohexamide, so basically cyclohexamide is a protein synthesis inhibitor, so it will not allow new protein to be synthesized. In that case, whatever MATS1 protein is already made, delta GR protein is already made, only that will enter inside the nucleus and it will activate immediate genes, but those genes which are getting activated at the transcription level, those RNA will not make protein because there is cyclohexamide protein, and if protein is not made, their target will not be activated. So, the MATS1 indirect target will not be activated, but direct target will be activated, and that is how you can see here when we treat dexamethasone and cyclohexamide together, you cannot see the activation of OSMGH3. This tells that OSMGH3 is not directly regulated by MATS1, but it is indirectly regulated by MATS. And then if you look this construct, this is another very important construct. So, this is a case when you down regulate MATS1 gene, what happens that it is all the floral organs are defective, you will not make a fertile seeds. You can see here this is the floral organs, they all are getting converted into leaf like structure or kind of lemmapelia like structure, you will not have organ. So, it is very challenging to go to the next generation, it is almost impossible. In that case, what you can do, you can make a MATS1 delta GR fusion as well as artificial microRNA, and then what you do, normally this artificial microRNA will suppress the gene, you have the phenotype, but if you grow in presence of dexamethasone, this delta GR will go and it will complement the phenotype. So, this construct has overexpression as well as artificial microRNA, but important thing here is that this artificial microRNA is designed to target 3' untranslated region of the MATS1, and that 3' untranslated region is not present here. Here we have taken MATS1, sorry, here we have taken MATS1 only till the stop codon, untranslated region is not present. Why? If we take that region, then this artificial microRNA will go and silence this gene as well. So, in this construct, this microRNA will specifically silence the endogenous copy of the gene, but it will not silence the transgene copy, and this way you can use a complementation based assay and do whatever phenotypic analysis, or you can go even identify the downstream target, direct downstream target to study it. You can see here when you induce with the dexamethasone, protein is getting localized to the nucleus, but when you do not induce, you do not see the protein inside the nucleus. Another very important technique which is being used to regulate both temporal as well as spatial expression simultaneously is XVE based system or estrogen receptor based system. So, here basically what has been done, so one chimeric transcription factor had been generated. So, this XVE is a chimeric transcription factor. Here there are 3 components, one is X, V and E. X is a DNA binding domain of LEX-A protein, LEX-A is from the bacteria, and then V is a transactivation domain of VP16 protein which is from virus, and then E is the human estrogen receptor. And when you combine this, so you have a kind of protein, fusion protein, which is a kind of artificial transcription factor which has a DNA binding domain and transactivation domain, but at the same time it is also fused with the delta GR. So, you can regulate its translocation from cytoplasm to the nucleus. And when you drive this expression under any promoter, if you choose a tissue specific promoter, you can drive this XV only in a specific tissue wherever you want. Why this is required? Because the previous delta GR based system you can use only for the protein which are functional in the nucleus. If you want to study a protein which is not functional in the nucleus, that technique might not be very useful. So, here what you can do, you can basically regulate both ways, and then what you do, you just put a Lex A binding site somewhere here just upstream to a basal promoter, and then put your gene of interest whatever you want to express. You want to express overexpression construct, you want to express RNAi construct, artificial micro R, whatever construct you want to express, you express here. Then what will happen here, normally this promoter will drive expression of the XV protein, but this protein will not enter inside the nucleus because of the estrogen receptor. But when you treat this with estradiol, 17 beta estradiol, this artificial protein will go and it has binding site here, it will bind here, and the transactivation domain of this transcription factor will activate the gene which is cloned downstream of it, and this can be used for even non-transcription factor kind of protein. So, this has been recently very detailed discussed and generated lot of gateway based construct which can be used for making this kind of study. But if you see how strongly this is happening, so if you look here, so here the promoter is G1090, which is again artificial promoter, very broadly expressed promoter, and it, so XV is driven by this promoter, and downstream gene which is cloned is the GUS. So, if you see, if you do not induce the plant, you cannot see GUS expression at all, but when you induce this with the estradiol at different time point, you can see very strong GUS expression here. This is the enlarged view of the root. You can see under mock treated condition there is no expression, but when you induce with the estradiol there is a very high expression. You can see here the functional significance. So, here what has happened, that XV has been cloned under G1090 promoter, and then plethora 5 is one of the trans AP2 domain containing transcription factor in Arabidopsis, and it has been cloned downstream of the Lex A binding site. And what happens when you do not induce, you see that your plant is normal, but when you induce with the estradiol, you can see lot of defect, developmental defect, which is consequence of overexpression of plethora 5. So, to conclude this, so we have now finished how to then once you have generated this transgenic, you can go and analyze its a phenotype as per your defined objective. So, now we will finish here, and then in next class we will study gene interaction, particularly genetic way or physical interaction or regulatory interaction. Thank you very much.